Okay, so now we're going to talk about bacteria pathogenesis. And so this is going to really be the beginning of us talking about bacteria and their infections. And so this is going to be a very big overview of pathogenesis before we get into individual bacteria pathogen. So with respect to pathogenesis, keep in mind that pathogens need to have virulence factors. And so the virulence factors are going to help the bacteria, in this case, or the pathogens, get into the patient, cause the disease, and be able to be transmitted. Now, for bacteria infections, we have two general classes of infections. We have our intracellular, and we have our extracellular. Now, our intracellular are bacteria that are pretty well known for causing infections. We have our mycobacterium and species. Of course, the famous one is going to be tuberculosis. But there are other ones that are constantly in our environment and do not cause trouble within us, but they would cause trouble if you have an immune compromised person. And then there's Listeria monocytogenes, there's Salmonella, and there's Shigella. And then for our extracellular bacteria, it's going to be a majority of them, but for the famous ones, we have our Streptococcus, and we have Staph, and we have E. coli, and we have Neisseria. And so one of the things about our bacteria infections is that we're going to have the diseases associated with them are going to be very different depending on what type of infection, obviously. And the immune response is going to be different, and the treatments are all going to be different. So treating an intracellular bacteria is very difficult. Treating an extracellular bacteria is easier. However, it can lead to a lot of damage to tissues. And so one thing that our gram-negative bacteria have are endotoxins. And so that's LPS. And then our gram-positive or negative can have exotoxins. And exotoxins are mostly going to be A, B toxins. And we'll talk about those. So now the stages of bacteria pathogenesis is going to be an infection, which then has a period of incubation. That leads to a prodrome. And then you have a specific disease. And then you have different outcomes. And so for our infection, this is where you have your virulence factors. Well, if it has a pellicle, if it has binding proteins, etc., adherence proteins. Our incubation is where you have the bacteria growth. Our prodrome is where you have your immune response, your acute phase response. Which is that IL-6, TNF, alpha, and IL-1. You also have, in this case, this would lead to, of course, fever plus nonspecific symptoms. And then, of course, you're going to have your specific disease. Is it respiratory? A GI infection? central nervous system symptoms. And then you're going to have outcomes. Outcomes are going to be recovery, which can have a long convalescence. It can have death, or it can become chronic. Now, just really quickly about exotoxins. We're going to talk about exotoxins in just a moment. Exotoxins are going to be relatively straightforward with respect to how they work. They're an AB toxin, and the A is going to be the active portion, and the B is going to be the binding. So this is the specificity. What targets the toxin? And keep in mind, an exotoxin can be toxic to the bacteria. So typically the bacteria will produce the exotoxin in an inactive form, and it doesn't get activated until it binds to its target, and then the active enzyme will be released and do its function. So again, exotoxins are secreted by gram-positive or negative. You're going to have the AB toxins. Here's some toxins that are very well known. We have our anthrax toxins, and this is going to increase cyclic AMP. And cyclic AMP increases are going to lead to localized edema as well as death to cells. We have Bordetella pertussis toxin. So there's a vaccine for it. And Bordetella pertussis toxin is going to also increase cyclic AMP. It does this through targeting adenylate cyclase, and then this is going to lead to cell death as well. Botulinum toxin, Clostridium botulinum, is going to decrease acetylcholine, and so you're going to get flaccid paralysis, and it's oftentimes going to present in the face first, so someone's going to look very droopy. Excuse me. Cholera toxin, Vibrio cholera, again, you're going to activate adenylate cyclase, and this leads to massive diarrhea, massive fluid loss, and if fluids aren't replenished quickly, the person can succumb to that loss. We have diphtheria toxin, Corynebacterium diphtheriae. This is going to inhibit protein synthesis, so this is a very toxic toxin. So inhibiting protein synthesis. Every cell in your body needs protein synthesis, except for your red blood cells, and so it's going to affect your body systemically. E. coli has several different toxins. One is the heat labile enterotoxin, and this is going to, again, activate adenylate cyclase, increase cyclic AMP, and cause diarrhea. We have pertussis toxin, which is going to be blocking G protein signaling. We have Pseudomonas exotoxin A, and this inhibits protein synthesis similar to E. coli. And Shiga toxin is also going to inhibit protein synthesis. So what's interesting is E. coli has a Shiga-like toxin. And so it's thought that E. coli acquired this enterotoxin from Shigella species. Now, Shiga-like toxin, I have it right here, for E. coli, does the same thing as Shigella toxin. And then tetanus, of course. Tetanus is going to have spastic paralysis, so it's different than our botulinum toxin. Botulinum toxin was flaccid paralysis. Tetanus toxin is going to be spastic paralysis. And so, again, just to point out, we have endotoxins. LPS is the only endotoxin. Sometimes they'll talk about Staph aureus. When we talk about Staph aureus, they'll have an endotoxin-like toxin, which we'll discuss that when we get there. But gram-negative bacteria always have LPS, and so they all carry with them this endotoxin. Now, this endotoxin, of course, triggers a toll-like receptor. And so, of course, this induces inflammation. And so it really depends on the amount present. That is why if you have a severe E. coli infection of your urinary tract infection, that's very serious because E. coli makes LPS, and it will release LPS, which will cause inflammation. And so LPS can lead to activation of our immune system, such as complement. It can lead to leukopenia, fever, intravascular coagulation, decreased circulation and perfusion to major organs, shock, and death. And so this can be a form of sepsis. So again, overgrowth of gram-negative bacteria, no matter what area of the body this is in, can lead to shock due to the release of endotoxins. And keep in mind that here's just a picture of what's going on, but LPS is going to trigger your immune cells. Your LPS is going to have effect on your platelets, on your mast cells to degranulate. Your macrophages are going to induce inflammation, and mast cells and macrophages are tissue resident, so they're going to be in every tissue in your body. So if you have a systemic release of LPS, you're going to have a systemic activation of mast cells and macrophages. And so again, this is just going to lead to overactivation of your immune system, overproduction of cytokines and different mediators of the immune response, and this overwhelms your body. Now, the last thing I just want to touch on are endospores. So endospores are going to be from gram-positive bacteria, and there's a few famous gram-positive bacteria that do produce these endospores. And we have our bacillus species, our serraceus, and our anthrus. And so they are going to form these endospores, and they are going to be potentially infectious. We also have our Clostridium. We have botulinum and tetani that will form endospores. And so the endospores are basically going to be little pockets of nucleic acids, and typically they have some proteins in there to help trigger when there's the right conditions for the endospore to germinate and become the bacteria.